Welcome to your channel, Silent Mind Studio, and thank you for listening to us. This is The Deep Dive.
Our mission, really, is to take a stack of sources, like the fascinating one we have today, and dive in head first.
Yeah, pull out those crucial nuggets of knowledge and insight. Think of us as guides, helping you get genuinely well-informed, fast.
And hopefully finding some surprising facts along the way, maybe sparking enough curiosity to keep you hooked.
Exactly. Today, we're embarking on a deep dive into something that sounds, well, profoundly futuristic. Animal minds, digital bridges.
We're talking about the science, the possibilities, and importantly, the ethics of connecting animal brains directly to external systems.
Maybe even someday to something as vast as the internet. It's kind of mind-bending stuff.
It really is. And the source material guiding us today is the text titled Animal Minds, Digital Bridges: The Science and Ethics of Neuro-Internet Connectivity in Non-Human Species.
Okay, so our goal for this deep dive, we want to unpack the science behind brain-computer interfaces, BCIs, especially how they're used with animals.
We'll trace the history, which is probably deeper than you think. We'll look at the tech feasibility, like, is a Neuralink for pets even realistic right now?
And then, crucially, we have to grapple with those big ethical questions.
Huge questions. Animal consciousness, consent, or the lack of it, the long-term impacts on species, our whole relationship with them.
If you are interested in content like this, we will discuss a topic that has intrigued us every day. Prepare for some pretty wild experiments and ideas.
Ideas that might make you rethink intelligence, connection, the works.
Definitely. Please be sure to subscribe to the channel and follow us so you don't miss future deep dives. We cover some fascinating ground.
Okay, let's jump in. Part 1, the technological frontier.
All right, right at the start, the source tackles the big one. What exactly is a brain-computer interface, a BCI?
Sure.
How does it actually work, basically?
Well, the source defines a BCI, or sometimes BMI, brain-machine interface, as a direct communication pathway.
Direct meaning?
Meaning it creates a link right between the biological brain and some kind of external device, like a computer. And crucially, it bypasses the body's normal way of doing things.
So it's not using your muscles, your nerves, anything like that?
Exactly. You're not thinking, move right, and then your hand moves the mouse. No.
Okay.
Instead, the BCI is designed to translate specific neural signals. These could be signals linked to an intention, maybe a mental state, or even just a learned pattern of brain activity.
And it translates that directly into commands?
Directly into commands for an action on that external device. The force breaks it down into 3 core steps, really. First, you got to acquire the neural signals.
Get them out of the brain somehow.
Yep. Then you analyze them, figure out what they might mean in that context, and finally you translate that interpretation into a command the system understands.
Acquire, analyze, translate. Sounds simple when you put it like that.
Conceptually, maybe. But the source immediately flags a major challenge, a kind of tension that drives a lot of the innovation here.
Oh, yeah. What's that tension?
It's the fundamental problem of getting a good signal from the brain. It's this constant trade-off. You want high fidelity, a clear, strong, detailed signal.
The best possible readout.
Right. But achieving that often means getting really close to the neurons, and that usually means surgery.
Ah, okay. Invasive.
Invasive versus the other side, which is developing systems that are safe and accessible. And that usually pushes towards methods that don't involve cracking open the skull.
And that's the core difference between invasive and non-invasive BCIs we hear about.
Exactly. That's the primary categorization the source uses based on how you acquire the signal. Invasive methods need surgery. You place electrodes right on the brain surface, that's electrocorticography, ECOG.
Okay.
Or you can go even deeper, putting tiny arrays of microelectrodes inside the brain tissue itself, intracortical microelectrode arrays.
Wow.
There are also techniques like CG, stereo EG, which target deeper structures, often used for things like epilepsy mapping, but potentially useful for BCIs too.
So why take those surgical risks? What's the payoff?
You get much, much better signal quality. Significantly better. Invasive methods give you higher spatial resolution. With those intracortical arrays, you can potentially listen in on individual neurons.
Single cells.
Potentially, yeah.
Yeah.
You also get a wider frequency bandwidth, so you capture faster brain activity up to 200 hertz, maybe even higher with ECOG. And the signal itself is stronger, the amplitude is greater, up to 100 microvolts for ECOG sometimes.
Why stronger? Just being closer.
Exactly. The electrodes are right there next to the action. You don't lose signal or get as much distortion from the skull, the scalp, all that stuff in between.
The source mentions Neuralink here. How does their approach fit? Is it ECOG intracortical?
Neuralink is a prominent example of an invasive approach, but with a kind of unique twist on the intracortical idea.
How so?
They use thousands of these incredibly thin, flexible polymer threads. Each thread has electrodes, and they use a robot to implant them.
A rope?
Yeah. The goal is super high density, listening to tons of neurons at once, while hopefully causing less damage than older, more rigid electrode arrays because the threads are flexible. But, crucially, it's still complex surgery.
And the downsides of that surgery, the long-term implants, the source talks about those too, right?
Oh, absolutely. They are considerable. You have the immediate risks of any brain surgery infection, bleeding, tissue damage, but then you have the long-term challenges inside a living brain.
What happens over time?
The brain sees the implant as a foreign object. It mounts an immune response, you get inflammation, and crucially, glial scarring.
Scarring around the electrodes.
Exactly. Glial cells basically wrap around the electrodes, encapsulate them. This increases the electrical impedance, makes it harder to pick up signals, and eventually it can degrade or totally block the signal.
So the implant stops working well or stops working entirely.
That's the risk. Maintaining biocompatibility, getting the body to accept the implant, and just the physical durability of the device over years and years is a massive engineering hurdle.
Okay, so invasive, top-tier signal, but big risks, big long-term challenges. What's the alternative?
The alternative is non-invasive BCIs, no surgery required.
Much safer, I imagine.
That's the huge advantage, safety and accessibility. This makes them way more practical for many potential uses, especially for animals where invasive surgery is a really high ethical bar unless there's a clear medical need.
Right. So what's the most common non-invasive method?
That would be electroencephalography, EEG, using electrodes placed on the scalp.
Swim cat-looking thing with all the wires.
That's the one. EEG's strength is its temporal resolution. It's very fast, great at telling you when things are happening in the brain, millisecond by millisecond. And the gear can be relatively portable.
But what's the catch?
The catch, as the source points out, is lower spatial resolution compared to invasive methods. You can't pinpoint activity nearly as precisely. And the signal-to-noise ratio is much worse.
Why worse?
Because that electrical signal has to travel through the skull and scalp before it reaches the electrodes. Those layers distort it, smear it out, weaken it. It's like trying to listen to a conversation through a thick wall.
Okay. Are there other non-invasive ways besides EEG?
Yes. The source mentions a few others. There's magnetoencephalography, or MEG. It measures the tiny magnetic fields produced by the brain's electrical currents.
Magnetic fields. Does that work better?
It actually offers better spatial resolution than EEG, and the skull doesn't distort magnetic fields as much as electrical ones.
Oh,
There's always a but.
The equipment is enormous, incredibly expensive, and you need a special room shielded from all external magnetic fields. So not very practical for everyday use.
Okay, what else?
Then there's fMRI, functional magnetic resonance imaging. It measures changes in blood oxygen levels, the BOL-D signal, which correlates with neural activity.
fMRI gives those amazing brain pictures, right? It's exactly. Fantastic spatial resolution shows activity across the whole brain. The downside, temporal resolution is poor. Blood flow changes are slow compared to neurons firing, so it's not great for real-time BCI control in most cases. Plus, the machines are huge and expensive.
Got it. Any others?
One more mentioned is FNIRS, functional near-infrared spectroscopy. It shines near-infrared light into the head and measures how much is absorbed by oxygenated blood near the surface in the cortex.
Infrared light, how does that compare?
It's more portable and much cheaper than fMRI, which is good, but it also had limitations. The temporal resolution isn't great, it can't see very deep into the brain, and the spatial resolution is also limited compared to fMRI or invasive methods.
So this fundamental tension, high-quality signal needing risky surgery versus lower-quality signals being safer and easier really shapes the whole field of BCI design.
It absolutely does, and it determines what kinds of interactions are even possible right now. For animals, especially diverse pets, the ethics of invasive surgery for something non-therapeutic are, well, really difficult.
Yeah, you wouldn't do brain surgery on your cat just for fun.
Hopefully not. So that pushes research towards either making non-invasive methods much better or developing invasive tech that's significantly safer and more stable long-term. It's also why there's so much work on new materials and using advanced software, especially AI, to try and pull meaningful information out of those weaker or noisier signals.
The source also mentions different ways the user interacts with the BCI mentally. It's not all just thinking move left.
Right, there are different modes. Active BCIs are what most people think of you consciously perform a mental task, like imagining moving your hand or doing mental math. The system learns to recognize the brain pattern for that task as a command.
Okay. Actively doing something in your head.
Then there are reactive BCIs. These rely on the brain's automatic responses to external things, like maybe lights are flashing at different speeds and you focus your attention on one. Your brain generates a specific response, like the P300 signal, something called SSVEP that the BCI picks up. You're reacting, not generating the command from scratch.
Interesting. Like using the brain's built-in reflexes almost.
Sort of. And the third type is passive BCIs. These are different again. They don't need you to do a specific task at all. They just monitor your background brain state.
Like what?
Like how focused you are, your cognitive workload, maybe even your emotional state. The idea is the system could then adapt passively, maybe adjust the difficulty of a task, or change the lighting in a room based on your detected state.
And you also mentioned the direction of information. It's not always brain out.
Exactly. Most BCIs we talk about are output, brain signals, control a device. But they can also be input or bidirectional. They can deliver information to the brain.
How? Like stimulating it?
Yeah, through electrical stimulation, potentially.
Yeah.
This could be used to try and restore senses, like creating artificial vision or touch for people who've lost them.
Mm-hmm.
Or it could be used to modulate brain activity, maybe calm seizures or treat depression.
And this is important for the animal context.
Very important.
Yeah.
Because it raises the possibility, even if it's way down the line and super speculative, not just of reading from an animal's brain, but of writing to it, influencing their perception, maybe their behavior. That opens up a whole different ethical can of worms.
Absolutely. And just to be crystal clear, the source defines a true BCI as using signals specifically from the central nervous system, right? The brain or spinal cord.
Yes, that's a key distinction. It separates BCIs from other interfaces that might use muscle signals or eye tracking or voice commands. It's about tapping directly into the CNS activity, which requires that higher level of tech and interpretation to really claim your connecting minds.
So understanding all these distinctions, invasive versus non-invasive, active versus passive, output versus input, why is that baseline knowledge so important when we start evaluating this idea of linking an animal's mind to, say, the internet?
Because it grounds the whole conversation. It tells you what's technically hard, what kinds of information we might realistically get or send, given today's limits. Are we talking basic on-off signals or something incredibly rich and nuanced? Almost certainly the former right now.
Right.
And it highlights that connecting a mind isn't like plugging in a USB drive. It's this complex, often noisy translation process, heavily mediated by algorithms.
Yeah.
And crucially, the level of invasiveness directly shapes the ethical debate, especially when you move from clear therapeutic goals to potentially elective uses in healthy animals.
That makes total sense. It's a system with layers and trade-offs and limitations at every step.
Okay, given this tech baseline, let's revisit the history.
Yeah.
You said it has surprisingly deep roots, even before the term BCI existed. Let's trace that path.
Yeah, the idea that the brain produces electricity and that maybe we could interact with it goes way back. The source points to the 19th century. A physician named Richard Caton in England reported recording electrical signals from the brains of rabbits and monkeys back in 1875.
1875. Wow. Yeah. And others like Adolf Beck in Poland, William Cybulski too, were doing similar work on animals around the same time.
So animal research was literally there from the very start of brain electrical recordings.
Absolutely foundational. The first actual recording that showed different types of brain waves, what we now recognize as maybe alpha and beta waves in animals, was done by Vladimir Protochninsky in 1913. He called it an electrocerebrogram.
Electrocerebrogram. Kitschy.
Maybe not. But the groundwork for human non-invasive recording came later with Hans Berger in Germany. He made the first human EEG recording in 1924, published it in 1930. He also identified alpha and beta waves in humans. That really opened the door for analyzing brain activity from outside the skull.
Okay, so early animal work, then human EEG. What happened mid-century? Did the tech get better?
It did. Techniques like ECOG, placing electrodes right on the brain surface, were developed, notably by neurosurgeons Wilder Penfield and Herbert Jasper in the 1930s. They were mainly using it during epilepsy surgery to map out where seizures started.
So an invasive technique, but giving clearer signals than scalp EEG.
Much clearer. And then in 1964, W. Gray Walter described something called slow cortical potentials, SCPs. These slow voltage shifts were later explored as a way for patients with locked-in syndrome, people completely paralyzed but conscious, to communicate.
And the 60s and 70s were pivotal again, especially for using animals in a new BCI-like way.
This is where it gets really interesting and directly leads to modern BCIs. The source highlights the work of Eberhard Fetz and his team at the University of Washington in 1969. This was groundbreaking.
What did they do?
They showed that monkeys could learn to voluntarily control an external device, a simple biofeedback meter, just by changing the firing rate of a single neuron in their motor cortex.
Oh, how did they teach them?
Operant conditioning. They rewarded the monkey whenever the neuron's activity went in the direction they wanted. It was the first really compelling proof that direct, conscious control of something external using only neural activity was possible. Other groups, like Schmidt and Baker, did similar work reinforcing this.
So an animal could literally learn to think in a specific way to move a needle on a dial, even using just one brain cell.
Essentially, yes. It was a cornerstone discovery, completely enabled by animal research. And it was building on this kind of work that led Jacques Vidal and his colleagues at UCLA, with NSF and DARPA funding in the 1970s, to actually coin the term brain-computer interface in their papers. Around the same time, M. Sturman was doing important studies on sensor-motor rhythms in cats.
Okay, so the term BCI emerged in the 70s. Moving into the 80s, was the focus still on just understanding the brain signals?
Very much so. The 80s brought huge insights into neural coding, how the brain represents information. Apostolos Georgopoulos at Johns Hopkins did really critical work with rhesus macaques.
What did he find?
He studied motor cortex neurons while the monkeys moved their arms. He discovered this elegant mathematical relationship, basically a cosine function, linking the firing rate of single neurons to the direction of movement. And this led to the idea of population vector coding. The understanding that movement direction isn't encoded by just one neuron, but by the combined weighted activity of a whole population of neurons firing together. That concept is absolutely vital for decoding movement intention in modern BCIs.
And the 90s seemed like things really started to accelerate towards practical applications.
They did. The 90s saw a big expansion and, critically, the first steps towards implanting BCIs in humans. A researcher named Philip Kennedy, who'd previously put special electrodes designed to encourage nerve growth into monkey brains.
Nerve growth into the electrode.
Yeah, neurotrophic cones. He performed the first invasive BCI implantation in a human patient in 1998. At the same time, Jonathan Wolpaw's group showed in 1991 that humans could learn to control a computer cursor just using their EEG signals, specifically the Mara rhythm over the motor cortex. And Niels Berbaumer in Germany was making progress using those slow cortical potentials for communication with paralyzed patients.
And the complex motor control BCIs moving robotic arms just by thinking that lineage comes straight from animal work in the 90s and early 2000s.
Directly. Big names here are Miguel Nicolas, John Donahue, Andrew Schwartz, Richard Anderson. They relied heavily on animal models, especially monkeys and rats, to develop and test the decoding algorithms and demonstrate sophisticated control.
That Nicolas internet experiment was famous, wasn't it?
Extremely. That 2000 study where an owl monkey's brain signals controlled a robot arm 600 miles away over the internet, even though it was initially open loop, no feedback to the monkey, it was a massive demonstration of potential. Later, they closed the loop, showing monkeys directly controlling robotic arms, predicting complex things like velocity and grip force just from brain signals.
And Schwartz had monkeys feeding themselves with robotic arms.
Yes. Andrew Schwartz's lab at Pitt had monkeys using a brain-controlled robotic arm to feed themselves fruit, showing really functional, dexterous control. John Donahue's BrainGate system came out of similar primate research at Brown, leading directly to the first human BrainGate implant in Matt Nagel in 2004, allowing him, a quadriplegic patient, to control a cursor and a simple robotic hand.
And Neuralink is just the latest iteration on this invasive path.
Neuralink, founded in 2016, is the most recent high-profile player. Their tech high channel count, flexible threads, robotic surgery represents the current cutting edge of invasive BCI development. And their first human implant in early 2024 was a major public milestone.
But it also brought a lot of attention to their methods, didn't it?
Yes, it brought huge market attention and invigorated research enthusiasm. But it also, as the source notes, came with significant public scrutiny and criticism, particularly around their animal testing transparency and practices.
Looking back at this history, it's just undeniable. Animal research wasn't just helpful, it was absolutely foundational to BCI development.
It's impossible to overstate. The initial proof of concept, understanding neural coding, developing decoding strategies, testing hardware. It all happened first in animals like monkeys, cats, rats. This deep reliance is exactly why the ethical questions around animal use are so central to the field.
And the history shows that push towards better tech, More stable, less intrusive.
Definitely. A constant drive for chronic, stable, less obtrusive systems, wireless, fully implantable. These are absolute prerequisites before you can even seriously think about something like a Neuralink for pets. And the entry of commercial players like Neuralink marks a new phase. Faster development, more investment, but also necessarily more intense ethical scrutiny.
Why does knowing this history matter so much for understanding where we are now?
Because it shows that today's cutting-edge tech didn't appear overnight. It's built on decades of painstaking scientific work, much of it relying on animals. It reveals the direct lineage from basic lab experiments to potential consumer tech. Understanding the origins helps appreciate the complexities and the ethical weight carried by this field.
Okay, we've got the basics, the history. Now let's get into the nitty-gritty. The engine room, as you called it. How do we get from that raw brain activity to actually controlling something? What are the core steps inside the system?
Right. The source breaks this down into that crucial three-stage process: signal acquisition, signal processing, and translation into commands and feedback. This is the core pipeline.
We covered acquisition methods, invasive, non-invasive, but remind us quickly about the types of signals they're trying to capture, especially with invasive methods. Why grab different kinds?
Good question. With invasive methods, especially those intracortical microelectrode arrays pushed right into the tissue, researchers can capture a few key signal types. They look for action potentials, spikes.
The actual neuron firing.
Exactly, the rapid electrical pulse when a neuron talks. They filter for these, typically between 300 hertz and maybe 12 kilohertz. They try to isolate single unit activity, SUA, listening to just one neuron. That's kind of the gold standard for detail, or multi-unit spikes, MSPs, from a small group nearby.
Okay, spikes. What else?
They also capture local field potentials, LFPs. These are slower electrical fluctuations, usually 0.5 to 300 hertz. They're thought to represent the summed synaptic activity, the inputs of thousands of neurons near the electrode.
So network activity rather than single neuron output.
Kind of, yeah. And specific frequency bands within the LFP are important, like high gamma LFPs around 7,300 hertz,
Yeah.
or the local motor potential, LMP, which seem to correlate well with movement. ECOG, from the brain surface, mainly picks up these stronger LFPs and can get good high-frequency signals up to 200 hertz or more.
And why record both spikes and LFPs? Do they tell you different things?
They seem to. Spikes give you very precise timing from individual neurons, their direct output. LFPs might capture broader network states, the overall level of synaptic input, and might be more stable over longer periods than tracking single specific neurons. Some newer methods like epidural field potentials, EFPs, recorded from the dura surface just outside the brain, might offer a less invasive way to get good LFP signals, especially high gamma. It's about capturing different facets of brain activity.
But the big advantage of invasive methods is still that superior signal quality. High amplitude, lots of detail.
Exactly. Better signal quality generally means more information, less noise, which leads to more accurate decoding of intentions or states. But, and it's a big but, those long-term challenges remain huge.
The biocompatibility problem.
Right. Surgical risks, cost, and especially the brain's reaction over time, inflammation, scarring, encapsulation degrading the signal, increasing impedance, potentially causing the implant to fail. That's why there's so much research into better materials, neurotrophic electrodes to encourage tissue integration, Neuralink's flexible polymers, conductive coatings like PDOT.PSS.
Yep.
But getting decades of stable recording from many neurons, still a really hard problem.
Okay, so step 1, acquire the signal with all its challenges. What happens in step 2, signal processing? The raw data isn't usable as is.
Not at all. Raw neural data is incredibly noisy and comes in huge volumes. Signal processing is essential to make sense of it. First basic steps. Amplification, because the signals are tiny, microvolts usually.
Boost the signal.
Yep. Then digitization, converting the analog electrical signals to digital data for the computer. Then a really important step, artifact removal.
Getting rid of the noise.
Exactly. Noise from muscles tensing, EMG, eyes moving, EOG, electrical interference from lights or equipment. Various filtering techniques are used here. But the absolute core of processing for BCI is feature extraction.
Feature extraction. What does that mean?
This is where the system identifies the specific patterns or characteristics within that cleaned up signal that actually correlate with what the user intends or the brain state you're interested in.
How does it find those patterns?
It depends on the signal. It might look at the amplitude of a brainwaves at a specific time point, like an event-related potential, ERP. More often, especially with EEG or LFP, it'll look at the power within certain frequency bands, like how much alpha, beta, or gamma activity there is. or specific rhythms like the sensor motor rhythm, SMR. It can also look at spatial patterns, which electrodes are active and how their activity relates.
And how are these features actually pulled out? What's the magic?
Algorithms, ranging from relatively simple mathematical filters and statistical methods to increasingly complex machine learning models, including artificial neural networks, ANNs.
AI is playing a big role here.
A huge and growing role. Yeah. For decoding movement, those population vector algorithms we mentioned are used. But AI is becoming critical for improving decoding accuracy from both high-resolution invasive signals and the much noisier non-invasive signals. The source notes that because the brain often uses large populations of neurons working together, just analyzing single neurons might not be enough for robust control. AI helps decipher those complex distributed patterns.
So it's less like listening to one specific voice and more like trying to understand the entire orchestra playing together.
That's a fantastic analogy. And AI is like the conductor or maybe the superpowered listener, helping make sense of that complex symphony.
Okay, acquired a signal, processed it, extracted the key features. What's the final step in the pipeline?
Step 3, translation into commands and feedback. This is where those extracted features get mapped onto specific commands for the external system.
Like move cursor left or select letter A.
Precisely. Or commands for a robotic arm, or even synthesizing speech. This mapping is usually done by classification or regression algorithms. And crucially, it needs to happen in real time for the BCI to feel useful.
And you mentioned feedback is critical here. Why?
Absolutely vital. Feedback closes the loop. It tells the user, human or animal, whether their brain activity actually produced the result they wanted.
How does it tell them?
It could be visual, seeing the cursor move correctly, auditory hearing, a beep for success, or even tactile, like a vibration. This feedback is essential because it allows the user's brain to learn and adapt through neuroplasticity. They learn to refine the neural signals they generate to get better control.
So you have to train to use a BCI.
Oh yeah. A training or learning period is fundamental for anyone or any animal to become proficient. It's not usually plug and play.
Okay, putting it all together. Connecting an animal's mind to the internet, if it were ever possible in a rich way, wouldn't be like a direct thought upload. It's this complex chain. Acquiring noisy signals, heavy-duty processing with AI to find patterns, translating those patterns into commands, and relying on feedback for the animal to even learn how to use it.
Exactly. The meaning isn't inherent in the raw signal. It's constructed by the algorithms and how they interpret those neural patterns. That's why validating what the BCI is actually decoding is so critical. The source also hints that maybe just looking at electrical signals is incomplete. Maybe we need multimodal approaches, combining electrical with chemical signals like neurotransmitters to get a fuller picture.
So when we hear buzzwords like mind connection, we need to remember it's built on these incredibly complex, challenging technical steps. Acquisition hurdles, noise, AI interpretation, and the absolute need for the user, animal, or human to learn and adapt. It's serious engineering, not magic.
Precisely. The technical depth and the inherent limitations are significant, even before we get to the philosophical questions about meaning.
Right. Okay, we've covered the tech basics. Now let's zoom back in on the animals. We know they were foundational historically, but let's highlight some specific pioneering studies. Which ones really showed what was possible using different animal models?
The source details several really key animal BCI studies that were milestones, demonstrating specific capabilities and providing essential insights.
Starting again with the non-human primates, the monkeys. They seem central.
They really are, given their brain complexity and similarity to humans. We have to revisit Eberhard Fetz's 1969 work again, because the source emphasizes it as absolutely fundamental.
The monkeys controlling the biofeedback meter.
Yes, proving that monkeys could learn volitional control based on single neuron activity. That direct brain-to-device link was shown to be possible. Then, Jugopoulos' work in the 80s with rhesus macaques on motor cortex population coding, figuring out the math behind how populations of neurons represent movement direction, provided the essential decoding principles for later motor prosthetics.
And then came the really sophisticated stuff, like controlling complex robotic arms.
That really took off in the late 90s and early 2000s, and primates were key. Miguel Nicoles's lab at Duke was incredibly influential here. They did extensive work with owl monkeys and rhesus macaques.
Including that internet experiment.
Yes, the 2000 owl monkey study controlling a robotic arm 600 miles away via the internet. Even though it started open loop, it was a massive proof of concept for transmitting neural data over distance. Later, they closed the feedback loop, allowing monkeys to see and directly control robotic arms in real time, decoding things like intended velocity and grip force with impressive accuracy just from brain signals.
Were other labs doing similar impressive motor control work with primates?
Well, yes. Andrew Schwartz's lab at the University of Pittsburgh had that famous work with monkeys controlling a sophisticated prosthetic arm in 3D to feed themselves pieces of fruit. That demonstrated really functional dexterous control via BCI. John Donahue's group at Brown used their BrainGate system in rhesus monkeys to track visual targets on a screen with a cursor, demonstrating precise closed-loop control with an invasive BCI that directly paved the way for the human BrainGate trials. And Richard Anderson's team at Caltech focused on signals from the posterior parietal cortex, an area involved more in planning movements.
Okay, so monkeys were absolutely crucial for figuring out how to decode complex movement intentions. What about decoding other types of brain activity? You mentioned decoding vision.
Yes, the source highlights some remarkable work done with cats. Yang Dan and her colleagues at UC Berkeley published a study in 1999 that caused quite a stir.
What did they do?
They managed to decode neuronal firing patterns from the cat's thalamus, specifically the lateral geniculate nucleus, a key relay station in the visual pathway, and use those patterns to reconstruct visual images the cats were looking at.
Reconstruct images, like see what the cat was seeing.
To some extent, yes. They could generate fuzzy but recognizable representations of natural scenes or moving patterns based solely on the recorded thalamic neuron activity. Garrett Stanley's group at Harvard did similar, very influential work around that time, too. It offered an unprecedented glimpse into how the brain internally represents sensory information.
That's incredible, decoding vision directly from brain signals. What about smaller animals like rats? Have they been important?
Rats have been workhorses in BCI research, especially for investigating underlying neurophysiology and for disease modeling. Companies like Aldigen Biosystems, mentioned in the source, have developed specific rat BCI systems.
For what purpose?
To study neurological diseases like Alzheimer's, Parkinson's, brain injury, spinal cord injury. They use implanted brain chips in these rat models to record neural changes associated with the disease and potentially test therapeutic interventions by delivering stimulation.
And this is also where the controversial rat cyborg research fits in.
Yes, that research largely falls under the rodent model work. Studies have explored creating rat cyborgs or robo-rats using implanted electrodes for remote control.
Remote control of their movement.
Exactly. Stimulating brain areas related to movement direction, often combined with stimulating reward pathways to reinforce the desired path. Proposed applications like search and rescue come up here, though, as we noted, this specific application is ethically fraught due to the direct control aspect. Rats are also used in more basic BCI research, like paradigms investigating how conditions like paralysis affect BCI learning and performance.
Have any larger animals besides primates been used in BCI research? Thinking about size closer to pets, maybe.
Yes. The source specifically mentions gutting in mini pigs. They've been valuable models for testing untethered, fully implantable BCIs.
Untethered meaning completely wireless.
Yes, systems with wireless power and data transmission. Pigs are a good model for evaluating the practical challenges of long-term stability, biocompatibility, and surgical techniques for these fully self-contained implants in a larger animal. Studies have demonstrated stable recording and stimulation in mini pigs for several months, which is crucial progress towards practical chronic BCIs.
Looking back at all these diverse animal examples, monkeys, cats, rats, pigs, what's the big takeaway about their collective contribution?
What really stands out is how each species offered unique advantages for answering specific critical questions. Primates for complex motor control, cats for sophisticated sensory decoding like vision, rodents for disease modeling, basic neurophysiology, and control principles. Larger animals like pigs for testing the engineering of long-term, practical, implantable systems.
So it wasn't random, it was targeted research using the best model for the question.
Precisely. These studies weren't just tech demos. They provided invaluable insights into brain function, how information is coded, brain plasticity, and the feasibility of interfacing technology with the nervous system. They show this clear progression from simple biofeedback to complex robotic control, even transmitting brain data across continents. And always pushing towards those more chronic, stable, wireless systems, the absolute prerequisites before you could even realistically talk about applications like a Neuralink for pets.
These animal studies really are the direct scientific ancestors of the technologies we're discussing today. They proved what was technically achievable in the lab, setting the stage for human trials and all the feasibility and ethical questions that follow.
They were absolutely indispensable for demonstrating the core concepts and capabilities that define the BCI field.
Okay, so who are the main players driving this forward now? Is it still mostly university labs, or have companies taken over?
It's definitely a mixed ecosystem now. You've got the academic institutions, significant government funding still playing a role, and increasingly very visible commercial ventures.
Academic labs are still the foundation, though.
Absolutely. They're the bedrock where much of the fundamental science, exploring new brain areas or decoding strategies, still happens. The source mentions key universities we've already touched on: Pitt, Hopkins, UW, Berkeley, Duke, Brown, Caltech, plus others globally like Tsinghua University in China, active in human trials. These places are crucial for discovery, developing new methods, training researchers, often funded by public grants.
And the commercial companies, what's their role? Just productizing the research.
They're becoming increasingly central, yes, translating research discoveries into potential products and services, but also driving development with significant private capital and engineering focus. Neuralink is the obvious elephant in the room right now.
Because of the high profile and Musk.
Exactly. Huge public attention, ambitious goals for both therapy and maybe enhancement. They've relied heavily on animal trials, monkeys, pigs, to develop their high-density flexible threads and robotic implantation. Their first human implant in early 2024 was a massive public milestone.
But that visibility came with downsides too, right?
Yeah.
Scrutiny.
Yes. The source explicitly notes Neuralink has faced considerable scrutiny and criticism regarding its animal testing practices and transparency. That's part of the landscape now.
Right.
Other commercial players are significant too. Altigen Biosystems, with their rat BCI systems for disease modeling and those rat cyborgs. Neural Signals, founded by the pioneer Philip Kennedy. Synchron is another major player, focusing on less invasive BCI tech for paralysis. BlackRock Neurotech is key because they make the research systems, like the widely used Utah Array, that many academic labs rely on for animal and human studies.
And government funding is still important in this mix.
Critically important, historically and currently. Agencies like DARPA in the U.S. have funded ambitious BCI programs for decades, often with military or restorative goals, but spurring broader progress. The NIH, particularly through the Large-Scale Brain Initiative, provides massive funding for basic neuroscience, BCI tech development, animal models, and significantly, neuroethics research. The NSF also funded early BCI work.
So you have this interplay. Universities doing basic science, governments funding priorities, and now companies trying to turn it into products with lots of capital. What happens when these forces collide?
It creates a really complex dynamic. Academic research lays the scientific groundwork. Government funding supports that and can steer it. Companies bring the investment, engineering scale, and regulatory know-how to try and translate it. This transition, especially with high-profile entries like Neuralink, shows the field is maturing. But it also introduces commercial pressures.
Like speed versus scientific rigor?
Potentially, yeah. The drive for speed and profit can sometimes clash with the necessary pace of careful scientific validation and deep ethical consideration, particularly around things like animal welfare and transparency, as some of the controversies around Neuralink illustrate. And the source notes this is global US, China, Europe are all major players. This brings both collaboration and intense competition and real challenges in aligning ethical guidelines and regulations across borders.
Understanding who's involved, universities, government, companies, helps you see the different motivations and potential pressures when thinking about where animal neurotech might go.
Exactly. It's not monolithic. It's a diverse landscape with different goals and incentives at play.
Okay, let's circle back to the why. Why use animals in BCI research in the first place? What are the different objectives researchers are trying to achieve with these studies?
The goals are actually quite varied, reflecting different scientific questions and potential applications.
What's been the biggest historical driver?
Historically, and still a primary focus, is restoring sensory and motor function, but for humans. Animal models are generally seen as an essential, often ethically or regulatorily required, step before testing neuroprosthetic devices in people.
So developing tech for paralysis, ALS, things like that?
Exactly. BCI research in animals aims to develop and refine systems to help people regain control. We talked about those landmark studies paralyzed monkeys controlling robotic limbs or wheelchairs just with brain signals. Those were critical proof-of-concept demos showing that human therapeutic solutions were potentially feasible.
Using animals as a necessary step towards human treatments. What other goals?
Another major one is modeling neurological and psychiatric disorders. Researchers use animal models, sometimes combined with BCIs or recording implants, to study the underlying brain mechanisms of diseases like Alzheimer's, Parkinson's, or spinal cord injury, and to test potential treatments in those models.
Like testing drugs or stimulation therapies in animal models of disease.
Right. The source also mentioned exploring models for things like chronic pain or psychiatric conditions. However, it's crucial to remember the caveat the source raises: the translational success rate.
The high failure rate when moving from animal models to human trials, especially for complex brain disorders.
Exactly. That's a significant challenge and ongoing debate in the field.
Is BCI research in animals also just about pure science? Understanding the brain?
Absolutely. A huge objective is understanding fundamental brain function. BCIs are incredibly powerful tools for basic neuroscience. They let scientists investigate how brains encode information, generate movement, process sensory input, support learning and memory, maybe even aspects of consciousness.
Like the cat vision studies?
Precisely. That work wasn't primarily about therapy. It was about understanding how the brain represents the visual world. Similarly, studies looking for neural correlates of sensory consciousness in birds, like crows, fall into this category, trying to find the neural basis of subjective experience in non-mammalian brains. This fundamental research often sheds light on human brain function too, due to shared evolutionary history.
Okay, human therapy, disease modeling, basic science. What about using the tech to directly change or improve the animals themselves?
That brings us to the more speculative and ethically charged objective: augmenting animal capabilities.
Augmenting, like making them better.
Potentially. This involves using neurotechnology to enhance or add sensory abilities, motor skills, perhaps even cognitive functions, often for purposes defined by humans. The rat cyborgs, remotely guided for search and rescue, fit here, augmenting their navigation for a human task. Conceptual ideas like interspecies brain-to-brain interfaces, maybe linking human senses with a dog's sense of smell, also fall under augmentation. This really shifts the focus from the animal as a model or subject to potentially altering its inherent nature for our benefit.
And finally, just using animals to test and improve the BCI technology itself, like a test bed.
Yes, that's another indispensable objective. Developing and refining BCI technology. Animal models are crucial for the iterative process of testing new hardware electrodes, implants, for evaluating biocompatibility, longevity, safety of new materials like flexible polymers, for refining surgical techniques, and for validating and improving the neural decoding algorithms. Using animals like the goating and mini pigs to test fully implantable, untethered systems is a perfect example, specifically tackling the practical engineering hurdles for long-term use.
So the goals span this wide range, from potentially curing human paralysis to basic science discovery to maybe creating super animals to just making the technology itself work better.
It's a good summary. The scope has definitely expanded from the initial strong focus on human therapy and basic science. Now it explicitly includes understanding animal cognition itself and this more controversial area of potential animal enhancement.
And that expansion raises the ethical stakes.
Significantly, especially when the goal shifts towards augmentation, primarily for human benefit, rather than the animal's own welfare. The risk-benefit calculation looks very different. The source also notes that decoding motor control is more established than decoding complex cognition or subjective states, and reminds us about limitations of animal models for uniquely human things and the translation issue. When the objective moves away from modeling humans towards understanding or modifying the animal itself, the ethical justification needs very careful scrutiny.
The purpose behind the research really matters. Is it for the animal, for humans using the animal, or something else? That framing seems key to the whole ethical discussion.
It's fundamental. Understanding the objective is the first step in evaluating the ethics.
Okay, let's really hone in on the core idea from the source's title, Connecting Animal Minds to the Internet. Based on everything we've discussed about current tech and research, how feasible is that specific idea right now? Is it realistic?
The source suggests that connecting animal minds directly to the Internet, especially in a way that conveys rich, meaningful information, particularly for diverse species like our pets, is currently much more speculative than real.
So science fiction for now.
Largely, yes. While individual components, BCIs, internet connectivity, exist in labs, reliably integrating them for this purpose, long-term, faces enormous hurdles.
What are the biggest technological roadblocks? You mentioned stability before.
That's probably number one. Achieving a truly stable, reliable neural interface for the long haul, especially with invasive implants. That foreign body response, the scarring, inflammation, encapsulation, degrades the signal, causes failures.
Even with new materials like Neuralink's threads.
Those aim to minimize the problem, and there's progress like months of stability shown in mini pigs. But achieving stable, high-quality recording from the same neurons or neuronal populations for the many years of a typical pet's lifespan, that remains a largely unsolved challenge.
So just keeping the physical connection working reliably over years is a massive issue.
It's a fundamental engineering problem. Then there's signal fidelity and resolution. Even if the implant stays physically intact, maintaining consistent, high-quality signal from the target neurons over time is hard. Brain tissue shifts, the tissue response changes. Single neuron stability is particularly tough, and non-invasive methods, still much lower signal quality, making decoding complex stuff very difficult.
And what about handling all the data that a high-density BCI would generate?
That's another huge one. Bandwidth and data processing. If you're recording from thousands of neurons at high speeds, you generate terabytes of data. Processing that, interpreting it, and transmitting it in real time for a continuous internet link. The demands are immense.
Didn't that early monkey internet experiment highlight this?
It did. It showed proof of concept, but also the challenges, transmission delays, managing the data volume. The source mentions the concept of an internet of animals in a different context, aggregating sensor data like GPS. That already needs sophisticated AI for management. A direct Neuralink would amplify this big data problem exponentially. You'd need incredible onboard processing in the implant itself to pre-process and compress data, or extremely high bandwidth, reliable wireless communication.
Powering such an implant long-term must also be tricky.
Definitely. Power requirements. How do you power a complex implant continuously for 10, 15, 20 years? Batteries have limited life, need replacing surgically, or are bulky. Wireless charging is promising, but has issues with efficiency, potential tissue heating, needing the animal to stay near a charger. Minimizing the power the implant consumes is critical.
And lastly, even if you get a stable signal, does the brain's code stay the same? Does the pattern for left always mean left?
That's the challenge of stability of recordings and decoding. Brains are plastic, they learn, they change. If the relationship between the neural patterns and the intended command drifts over time, the BCI needs frequent recalibration.
Which would be impractical for a pet owner.
Totally impractical, and potentially really stressful for the animal if the interface becomes unreliable. While some signals like LFPs might be more stable over the long term than single spikes, achieving truly consistent, reliable decoding over years without major recalibration is still an open problem.
So even just the basic infrastructure, the stable, long-lasting, high-bandwidth, low-power, reliably decoded connection itself, is still facing massive engineering hurdles, let alone piping that into the Wild West of the internet.
Correct. The core challenge is engineering this durable, reliable, wetware-hardware interface that works seamlessly within a living, changing biological system for extended periods. And the data volume requires huge leaps in processing, transmission, and AI interpretation for any meaningful real-time interaction over a network.
Before we even worry about what the animal's mind might say online, the technology just needs to reliably get the signal out and make sense of it for years on end. And the sources show that's far from solved for widespread long-term use.
That reliability and longevity is the absolute foundation, and it's not quite there yet for the scale and duration required for something like a mass-market Neuralink for pets.
Okay, let's play devil's advocate. Let's imagine, hypothetically, we solve those engineering problems. We get a stable, long-term signal. What about the next hurdle? Maybe the biggest one, translation. How do we actually know what the animal is thinking, feeling, perceiving from that blizzard of neural data?
This translation problem, as the source emphasizes, is perhaps the most profound and least understood part. It's not just about the physical connection. It's about deciphering the meaning encoded in the neural activity.
What can we decode with some success now?
As we've discussed, there's been significant progress in certain areas. Motor intent and movement is the most advanced, especially in primates. We can decode signals from motor cortex, parietal cortex to control cursors, robotic arms, wheelchairs with reasonable accuracy, often by looking at the activity of large neural populations. That's relatively well established for planned actions.
Okay, movement. What else?
We've made some inroads into decoding sensory information. We talked about the cat studies reconstructing visual scenes from thalamic activity. And more recently, that research on corvid birds, like crows, finding neural correlates of sensory consciousness linking single neuron activity to whether the bird reported seeing a stimulus or not. That suggests we might be able to find neural markers for basic perceptual states.
Decoding what an animal intends to move or roughly what it's seeing. That's definitely impressive. But it feels miles away from knowing if my dog loves me or what he's dreaming about or why he's anxious.
Exactly. And the source is very clear on this point. The leap from decoding specific motor plans or basic sensory inputs to translating the full richness of an animal's cognitive and subjective world, their thoughts, emotions, memories, consciousness, is immense. The challenges are profound.
What makes translating those deeper states so much harder?
Fundamentally, it's the sheer complexity of cognition and subjective experience. Things like learning, memory, decision-making, complex emotions, social understanding, consciousness itself, self-awareness. These are incredibly complex and, frankly, poorly understood even in humans, let alone across the vast diversity of animal species. Each species has its own unique sensory world, its umwelt, its own cognitive specializations shaped by evolution.
So translating the neural code for these is just vastly harder than for, say, move arm left.
Immensely harder. Plus, we face the other minds problem. We can only infer subjective experience in others, especially nonverbal animals, based on behavior and physiology. That inference is highly susceptible to misinterpretation and projecting our own human experiences onto them, anthropomorphism. Animal models also have limits for studying uniquely human things like language or abstract reasoning, accessing the animal's actual subjective state, largely beyond our current science.
Are there other ways researchers are trying to understand animal minds, maybe without sticking electrodes in?
Yes, there's a really interesting parallel field using AI and machine learning, but applied to natural animal communication, analyzing complex patterns in vocalizations, body language, facial expressions.
Like trying to build Google Translate for animals.
That's the analogy often used. Projects like the Earth Species Project are trying this with whales, prairie dogs, marmosets. They're looking for meaningful patterns in communication signals. Now, this doesn't use neural interfaces, but success here could potentially give us some ground truth to help validate interpretations of neural signals related to communication or internal states. But it's a separate, challenging effort itself.
What about decoding more abstract concepts directly from the brain, like thoughts about food versus danger?
That's called semantic neural decoding. It's being explored in human BCI research, trying to identify brain patterns linked to conceptual categories. For example, telling someone is thinking about tools or animals. Applying this to animals would require understanding their conceptual world and how it's represented in their brains, which is, again, largely uncharted territory.
And even if we could detect a signal for hunger, how do you translate that usefully for something like an internet connection? It's not straightforward, is it?
Not at all. That's the translation problem itself. Let's say you reliably identify a neural pattern linked to hunger. What information do you transmit? Raw data. A simple hungry alert. A more complex message. The source stresses that the meaning isn't just there in the signal. It's constructed by the algorithms and how humans or AI interpret it. This interpretation layer is critical and ripe for potential misreading or oversimplification.
So we might detect a pattern correlated with stress, but that's a universe away from translating the animal's actual experience of stress or the reason for it. And ultimately, humans are deciding what that pattern means.
Precisely. The gap is huge. AI is great at finding patterns, but validating what those patterns mean in terms of complex internal states in another species is the core scientific and philosophical challenge. The source notes that current Internet of Animals ideas mostly aggregate observational data GPS, activity levels, not direct internal neural states. That distinction is critical.
The tech might eventually link the brain physically, but truly understanding and translating the mind within, that's a whole different, much harder problem we haven't solved.
It's the difference between recording electrical activity and truly comprehending subjective experience and complex cognition. Miles apart currently.
Okay, this brings us squarely to the concept that really grabs headlines, a neural link for pets. Taking this BCI tech and putting it into our dogs, our cats. Based on everything the source says, what's the real-world status of that idea?
The source treats the idea of a widespread, sophisticated neural link for pets as highly speculative, certainly for the near future, even though it clearly builds on the decades of animal BCI research we've been discussing.
What's the scientific foundation that makes people even consider it? Why isn't it pure fantasy?
It rests on all that foundational work, mainly in primates, rodents, cats, showing it's possible to record brain signals, decode motor and sensory information, control external devices, and achieve some level of long-term implantation, despite the challenges. Neuralink's specific tech, The high-density flexible threads, the robotic surgery aiming for scalability and reduced damage, is often seen as pushing the engineering closer to something potentially adaptable.
Like the monkey playing Pong with its mind, that kind of demonstration.
Exactly. That's a high-profile example of the state-of-the-art in invasive BCI control in primates. The concept is essentially, could we adapt this advanced invasive tech or maybe future non-invasive breakthroughs for use in companion animals?
And if we could overcome the hurdles, what are the potential applications people dream up? The exciting, maybe slightly sci-fi possibilities.
The speculative potentials are pretty broad. The one you hear most is enhanced human-animal communication.
Finally knowing what Fido is thinking.
That's the dream, right? Translating neural signals into understandable information about needs, desires, emotional states, supposedly going beyond barks and meows. It could range from simple alerts, I'm hungry, I'm in pain, to, much more speculatively, richer insights into their internal world.
What else?
Other ideas include remote monitoring and interaction, checking on your pet's mental or physiological state via an internet-connected BCI, maybe interacting remotely, though how is usually undefined. Control of environment or assistive tech is another pet's, using thoughts to operate smart feeders, doors, or maybe assistive devices if they have mobility issues.
The source also mentioned therapeutic uses as more realistic.
Yes, therapeutic applications for pets are seen as the most ethically justifiable near-term potential. Using BCIs to help pets with severe neurological issues like epilepsy or paralysis, basically adapting the goals of human neuroprosthetics to veterinary medicine.
And the really far-out idea of making pets better somehow.
That falls under cognitive or sensory augmentation. It's more distant, definitely ethically contentious. Drawing parallels with human enhancement debates, it's the theoretical idea of directly augmenting a pet's senses or cognitive abilities, maybe giving them new inputs via the BCI.
Okay, those potentials sound incredible, like straight out of science fiction. But the source brings us back down to earth with a long list of practical problems. What are the major real-world challenges that make a widely available Neuralink for pets unlikely anytime soon?
The source emphasizes that the path from controlled lab experiments in specific species to safe, effective, affordable, and ethical devices for the millions of diverse pets out there is incredibly steep and fraught with practical hurdles.
Starting with the pets themselves, they're not all lab rats or monkeys.
Exactly. Species variability is a massive one. Think about the huge diversity just among common pets. Dogs of all sizes, cats, birds, rabbits, hamsters, reptiles. Their brain sizes, structures, neurophysiology, cognitive abilities differ enormously from each other and from the standard lab models like macaques or rats. Adapting the technology, the surgery, the electrodes, the decoding algorithms for each species would require immense dedicated R&D. You guys should take a primate BCI and stick it in a poodle.
And the cost. Brain surgery isn't cheap.
Cost and accessibility. Invasive neurosurgery and the sophisticated hardware are currently extremely expensive. The costs involved in developing, manufacturing, implanting, and crucially, maintaining these devices for the consumer pet market would almost certainly be prohibitive for the vast majority of pet owners worldwide. Non-invasive options might be cheaper, but as we discussed, they lack the signal quality for complex applications like nuanced communication.
What about the ethics of doing this to healthy pets?
Ethical and welfare concerns are profound. The source raises serious questions about the justification for performing invasive brain surgery on healthy companion animals for purposes that aren't essential for their health or survival. The risk-benefit calculation is vastly different from therapeutic use. The controversies around Neuralink's animal testing highlight the intense public and ethical scrutiny any company trying to market widespread pet implants would inevitably face. Ensuring long-term animal welfare, dealing with potential side effects or device malfunctions over a pet's lifetime is a huge responsibility and hard to guarantee.
What happens if the implant breaks or needs updating years later? Pets live a long time.
That's the issue of durability, maintenance, and long-term support. Pets are active, they tumble around, they get into things. Implants would need to be incredibly robust. But complications like infection, the device moving, or hardware failure are still significant risks over a 10, 15, 20-year lifespan. Managing these requires specialized veterinary care and technical support that likely doesn't exist at scale. Who provides that long-term support? It's already a major concern for human neurotech implants.
And we're back to the translation problem again, but now applied to all the different kinds of pet minds.
Yes, data interpretation and validation remains a monumental challenge. Accurately decoding complex thoughts, emotions, or intentions across the huge diversity of pet species, each with their unique cognition. It's an enormous scientific hurdle. Without extremely robust validation methods, The risk of misinterpretation and anthropomorphism is incredibly high. We might end up with flawed communication that reflects our own assumptions more than the animal's reality.
How would the animal even cope with having this thing in its head?
That's the concern about cognitive and behavioral adaptation. The animal would need to learn to use the BCI. What's the cognitive load? Could it be confusing or frustrating? What are the potential long-term behavioral changes from having a constant neural interface, especially one that might malfunction or provide ambiguous feedback? These are significant welfare unknowns.
So it really sounds like the idea of a rich, nuanced Neuralink for pets connecting minds to the internet is firmly in the realm of science fiction for the foreseeable future. The hurdles engineering for diverse species, cost, long-term stability, and safety, and our fundamental inability to truly interpret complex animal thought seem overwhelming right now.
That's a very accurate summary of the source's perspective. The technological leap required to go from controlled lab experiments in a few species to safe, effective, affordable, and ethically sound devices for the millions of diverse pets worldwide is vast. The most plausible near-term applications are likely limited to specific, high-need therapeutic uses in veterinary medicine. Adding the internet connectivity layer for complex communication just adds even more layers of technical and ethical complexity: data privacy, security, potential for remote manipulation, psychological impacts that are even further from being solved.
It's a compelling, almost seductive idea, but the sources really highlight just how many fundamental problems need solving before it could ever become a reality, especially for the animals we share our lives with.
The vision is powerful, but the practical and ethical barriers are currently immense.
Okay, let's pull back from the cutting edge again. You mentioned earlier that connecting animals and technology isn't new. How does neurotechnology, this idea of brain interfaces, fit into the much longer history of humans using tech with or on animals?
You're absolutely right. It's not new at all. Neurotech is really just the latest and maybe the most intimate chapter in a story that goes back thousands of years. It's part of the long history of how humans have integrated animals into their technological development.
Starting way back with domestication itself.
Exactly. The source frames domestication as the earliest phase. It wasn't just taming, it was actively altering species evolution through selective breeding to serve human needs: food, labor, materials, companionship. And alongside that came the invention of technologies specifically for managing and controlling those animals. Things like yokes, bits, bridles, fences, enclosures. Technology was used on animals to shape their lives for human purposes from the very beginning.
So technology has always mediated that relationship.
Fundamentally. And as societies and tech evolved, so did the ways animals were integrated. They became subjects of study, tools for research, sources of data. Early tracking methods were essentially rudimentary tech applied to animals.
Like bird banding.
Yeah, John James Audubon did bird banding back in 1803. Hans Christian Mortensen systematized it in the late 1800s. Even things like scale clipping on reptiles, noted from 1933. These were simple technologies, but they extended our ability to observe and gather data from animals.
And what about technology for animal health? Veterinary medicine.
That evolved too, using specialized instruments for animal bodies. Even in Roman times, surgical tools were adapted. The 19th century saw more specific veterinary tools, like tracers, emasculators, plus advancements in restraint techniques before anesthesia was common. And animal prosthetics have a surprisingly long history.
Prosthetics for animals, like wooden legs.
Literally, yes. The source mentions 19th century Britain fitting wooden legs to valuable working cattle. The motivation there was purely economic, maintaining the animal's productivity. Later, as veterinary care professionalized and pets became more valued emotionally, you saw things like glass eyes or false teeth for companion animals.
But the underlying theme seems to be human benefit driving the tech application.
That's a critical point the source emphasizes. Whether for labor, science, economics, or even sentiment, the historical use of technology on animals was overwhelmingly driven by human needs and perspectives. This long history of instrumentalization is the essential backdrop for today's ethical debates about neurotechnology, which represents a potentially far deeper level of intervention.
And animals became central to scientific discovery itself, beyond just observing them.
Absolutely. While early naturalists observed the rise of experimental psychology and physiology in the 19th and 20th centuries, really positioned animals as models for understanding biology and behavior under controlled conditions. Think of Pavlov's dogs, Lavoisier's guinea pigs for respiration studies, Pasteur's work on vaccines using sheep, Thomas Hunt Morgan's fruit flies revealing the basics of genetics. These discoveries relied fundamentally on animal experiments.
And the scientific use led towards directly interfacing tech with their bodies and brains.
Yes, that trajectory culminates in the widespread use of animals in biomedical research today. They became indispensable for understanding disease, developing drugs, testing safety, and this is where bioinstrumentation became standard, directly interfacing animal bodies and, crucially, their nervous systems with recording and stimulation technology. Modern neuroscience and BCI research specifically grew directly out of this practice. Early neuroscience work, like Galvani showing electrical stimulation made frog legs twitch, or Romoni Cacciao mapping neurons using microscopes on animal tissue, paved the way conceptually.
So there's a clear historical line from simple electrical recordings in the 19th century to the complex primate BCIs decoding movement today.
A very clear line. It shows a trend towards ever more intimate and sophisticated technological integration with animal nervous systems. And this increasing sophistication naturally intensifies the ethical questions about animals as living instruments. Moving from physical tags to implantable electrodes represents a shift towards more direct, invasive interaction, setting the stage for the profound questions raised by neuro-internet connectivity.
Wow. It's powerful to see how neurotech isn't appearing in a vacuum, but as the latest step in this very long history of human-animal technology interaction, mostly driven by human needs.
It provides crucial context. While the tech is novel, the dynamic of humans applying technology to animals for human purposes is ancient.
Building on that historical role, the source strongly emphasizes how pivotal animal models have been in advancing both human and veterinary medicine. What are some key examples of breakthroughs cited?
The source underscores that their impact is undeniable. Animal models have been essential for investigating basic physiology, understanding how diseases work, and for testing the safety and efficacy of countless therapies and technologies. Much of modern medicine rests on knowledge gained from animal research.
Can you give specific examples of major medical wins that relied on animals?
Absolutely. The source lists several critical ones. The development of the polio vaccines, which nearly wiped out the disease globally, heavily relied on research using rhesus monkeys. Techniques for successful organ transplantation were largely developed and refined in animal models first. The discovery of insulin as a treatment for diabetes came directly from experiments involving dogs.
Insulin from dogs, wow.
Yes, lithium for treating bipolar disorder was identified through studies in guinea pigs, and the development of safe, modern general anesthetics, which revolutionized surgery, also required crucial animal studies. We also mentioned Thomas Hunt Morgan's foundational work on genetics using fruit flies understanding chromosomes and inheritance.
Those are truly transformative medical advances.
They are. And in neuroscience specifically, animal models remain indispensable for studying basic brain function, the neural basis of behavior, and the pathology of devastating neurological and psychiatric disorders like Alzheimer's, Parkinson's, stroke, and spinal cord injury. And as we've established, BCI development itself has been profoundly reliant on iterative animal experiments, especially using primates for complex motor control research due to their brain similarities and dexterity.
So the positive impact on human health is undeniable and massive. But the source also flags limitations and challenges, particularly with translating findings from animals to humans. What does it say about that side of the coin?
Right, the source is careful to point out that while historically crucial, the utility of animal models isn't limitless, especially when it comes to translatability. There's growing evidence and concern about the high failure rate of treatments that look promising in animal models but then fail in human clinical trials. This is particularly true for complex conditions like neurological and psychiatric disorders.
What kind of failure rate are we talking about?
The source cites figures suggesting around 90% of drugs that succeed in animal studies ultimately fail in human trials. For Alzheimer's disease treatments specifically, the success rate translating from animal models to humans is reported as shockingly low, maybe only 0.4%.
0.4%, that's incredibly low. Why such a big gap between animal results and human outcomes?
The reasons are complex and debated, but include fundamental biological differences between species in brain structure, genetics, immune responses, how drugs are metabolized. Also, many animal models of disease are necessarily artificial. They might mimic some aspects, but often don't fully replicate the complexity, cause, or progression of the human condition. And there are ongoing critiques about the validity of some models, especially for psychiatric disorders. Do they really capture the human experience?
Are these translation problems leading researchers to look for alternative methods? Ways to reduce reliance on animals?
Yes, definitely. These translational challenges, combined with growing ethical concerns and the sheer cost of animal research, are driving a major push towards developing and validating what are called new approach methodologies, or NAMs.
And names, what are those?
And NAMs cover a range of non-animal techniques. Advanced in vitro methods are a big part using human cells to create things like organoids, including complex brain organoids or brains on a chip, and microphysiological systems, organs on chips, that try to replicate human tissue function in the lab. There are also in silico approaches, sophisticated computer modeling, machine learning, AI to simulate biological processes or predict drug effects, and in chemical methods too.
And AI is seen as potentially helping develop these alternatives.
Absolutely. AI is viewed as a powerful tool for analyzing complex data from NAMs, potentially identifying patterns relevant to human disease, and ultimately helping to reduce reliance on animal testing in areas like neurology research. Brain surrogates like organoids or even chimeras using human cells and animal embryos are being explored for studying human brain development with potentially greater accuracy. Although the source notes these raise their own unique ethical questions.
Is this shift away from animal models happening quickly, or is there resistance?
The source suggests there's momentum, but also significant inertia. The long history of success with animal models has built up strong precedent, infrastructure, and perhaps even bias. There might be an animal methods bias in how research gets funded or published, potentially slowing the adoption of promising NAMs, even when translatability is a known issue.
It seems almost paradoxical that the BCI technology developed using animals might actually help create tools like better ways to study organoids that could eventually replace animal use in some areas.
It's a fascinating feedback loop. Technologies refined through animal BCI work, sophisticated neural recording, data analysis, maybe even BCI-controlled organoid systems could paradoxically contribute to the goals of replacement, reduction, and refinement, the 3 Rs, by providing richer, potentially more human-relevant data streams from non-animal systems.
And does that high failure rate in translation affect the ethical justification for using animals in the first place?
It absolutely fuels the ethical debate. If the likelihood of a treatment developed in animals actually benefiting humans is very low for certain complex diseases, then the benefit side of the harm-benefit analysis used to justify the research becomes weaker. This makes the harm cost to the animals harder to justify from a utilitarian standpoint, increasing the ethical pressure to find more predictive alternatives.
So it's this complex picture. Animal research has been undeniably foundational and led to huge medical gains, but we're also increasingly aware of its limitations for translating to complex human conditions, which is driving the search for alternatives.
It's a very dynamic landscape. The historical utility is clear, but the translational hurdles for some major diseases, combined with ethical pressures, are pushing the field towards new approaches, some of which, ironically, leverage the very technologies that animal research helped create.
Understanding this context, the successes, the limitations, the push for alternatives is crucial for navigating the ethical tensions around animal neurotech today.
Absolutely vital background.
Okay, let's shift focus again. Moving beyond just using animals as models for us, the source talks about neurotechnology potentially acting as an artificial evolutionary force, actually changing the animals themselves. How could this tech enhance or augment what an animal can do?
This is where we step into a potentially transformative and ethically complex new phase. The idea is that neurotechnology enables direct technological modification of animals, potentially redefining them and their role in the world. The source suggests this could indeed act like an artificial evolutionary force, driving changes much faster than natural selection.
What are some specific ways this augmentation could happen?
One major pathway is sensory augmentation, using neural interfaces to enhance existing senses, or maybe even give animals entirely new ones.
Like giving them superpowers.
Sort of. The source mentions rat BCI research, exploring the possibility of enabling rats, which normally can't see it, to perceive infrared light. They did this by converting infrared signals into electrical stimulation delivered to the part of the brain that processes touch, somatosensory cortex. The rats learned to use this new sense.
So the principle is established. You can feed new sensory data into the brain.
The principle seems feasible, yes. Feed novel sensory information into the appropriate brain area via a neural interface to alter or extend perception. This could also include those interspecies concepts, like maybe co-opting a dog's amazing sense of smell and feeding that data back to augment human olfaction.
What about physical augmentation, improving movement?
Motor augmentation and restoration is another key area, and probably the most developed technically due to the therapeutic research. We've talked about the primate studies controlling prosthetic limbs, cursors, wheelchairs via motor cortex signals. While often framed as restoration for injury, it clearly demonstrates the capacity to extend an animal's physical reach and interaction capabilities through neurally controlled machines.
And could we use this tech to make animals smarter or change how they think?
That's cognitive augmentation, which is much more speculative but definitely discussed, drawing parallels from human cognitive enhancement research. Could we directly enhance learning, memory, or problem-solving abilities using neural interfaces? We know animal brains or plastic environmental enrichment changes them. Direct neural stimulation, part of bidirectional BCIs, could theoretically target brain regions involved in learning or memory to try and boost function. But this is highly ethically charged.
The ability to directly stimulate the brain seems key to these augmentation ideas, but also risky.
Very risky. Modulation of brain activity via stimulation is powerful. It's been shown in rat BCIs to induce desired behaviors and in mini pigs using wireless stimulation, but it carries a significant risk of coercive control.
Forcing the animal to do something.
Exactly. Versus genuinely beneficial shaping or enhancement. This raises huge welfare and autonomy concerns.
The idea of directly enhancing senses or controlling movement remotely, or maybe making animals learn faster via brain implants, it's pretty striking. But the source mentions this blurs fundamental lines. What lines?
Yes, it blurs several lines. If you fundamentally augment an animal's capabilities, you alter its ecological niche, how it interacts with its environment, its relationship with us. You could disrupt natural social dynamics. If some individuals have technologically enhanced senses or communication, you might create new dependencies. And the line between therapeutic restoration, a prosthetic leg for an injured dog, and enhancement purely for human benefit, like creating a super sniffer dog for security, is often very blurry and ethically contentious.
And animals can't consent to becoming enhanced.
Precisely. The instrumentalization becomes much more profound when we're talking about altering their fundamental biological capabilities for our goals. We're moving beyond using them as models to potentially reshaping their very being.
This isn't just about reading brain signals anymore. It's about the potential to actively change what an animal is, what it can do. That raises huge questions about their natural state and who gets to decide what an animal should be.
It represents a potential future where humans act as direct engineers of animal capabilities via technology, that artificial evolutionary force. It demands extreme caution and deep ethical reflection because the consequences could be immense and irreversible.
Let's go deeper into those really radical possibilities. The source touches on concepts like cyborgs and linking multiple brains. What does the source mean by technologies that are blurring biological boundaries?
This refers to neurotechnologies that really push against our standard understanding of individual biological organisms, either by turning a living being into a directly controlled tool or by merging the function of multiple brains.
Tell us more about the rat cyborgs or robo-rats. That sounds like something from a movie.
It does, but it's based on real research, including work by companies like Altogen Biosystems mentioned in the source. These are animals, typically rats, implanted with electrodes that allow for remote control of their movement and navigation.
How does that work? Zapping their muscles?
No, directly stimulating the brain. Specific areas in the motor cortex might be stimulated to make the rat turn left or right. And often, reward centers in the brain, like the medial forebrain bundle, are stimulated to reinforce the desired path and guide the animal through complex environments, like rubble.
So they're steering the rat with brain stimulation. What's the proposed use?
Applications like search and rescue in disaster zones or potentially military reconnaissance have been suggested.
This sounds incredibly controversial.
It is, highly. The ethical debate centers squarely on animal welfare, autonomy, and extreme instrumentalization. Researchers might argue the reward stimulation isn't painful, but the core ethical objection for many is the profound loss of agency, the act of direct, external control over a living, sentient being's locomotion, essentially treating it like a biological drone.
Okay, that's controlling one animal. What about brainettes, linking multiple brains?
This is even more conceptually radical, pioneered by Miguel Nicolelis' lab at Duke. They've demonstrated the feasibility of linking the brains of multiple animals together using BCIs to form a computational network they termed a brainette.
How on earth did they link brains together?
In their early rat brainette experiments, they interconnected the brains of several rats via BCIs. The rats learned to synchronize their neural activity in real time to achieve a shared goal, like all pressing a lever at the same time to get a reward. Interestingly, they found the group, the brainette, performed the task better than the individual rats could alone.
So the linked brains were more capable together.
In that specific task, yes. In an even more striking follow-up, they created a serial processing rat brainette. Information like a sensory stimulus given to the first rat was processed sequentially through a chain of 3 interconnected rat brains. The brain activity in the third rat could actually predict the original stimulus given to the first rat.
Wow. So the chain of brains was performing a computation.
Essentially, yes. It demonstrated that the brainette could perform basic computational tasks like information storage, transfer, even pattern classification. The source notes it suggests the animal's sensory cortices were acting like biological processors in a network, rather than thinking together in a conscious way.
Did they try this with primates too?
They did. In separate experiments, they linked the brains of 3 rhesus monkeys.
Yeah.
Each monkey used its BCI to control 2 of the 3 dimensions needed to move a virtual arm on a screen. They had to work together.
And could they?
Yes, they learned to synchronize their neural activity and collaboratively perform the 3D movement task. The system even showed robustness. If one monkey briefly disengaged, the other 2 could often compensate and keep the task going. It's important to note these monkeys weren't physically wired together like the rats. Their brains were linked functionally through their individual BCIs and the shared task structure.
So not just one animal interfaced with a machine, but multiple biological brains, potentially even across species in theory, linked up and functioning as a collective system. This really does blur the lines of individuality.
It signifies a major conceptual shift. It's about creating hybrid cognitive systems from multiple biological brains or integrating brain functions across species. And this raises entirely new ethical questions.
Like what?
What's the moral status of an emergent brainette that has capabilities beyond its individual parts? Who is responsible for the welfare of this distributed entity? The instrumentalization is stark individual brains, used as nodes or effectors in a larger system, usually for human goals. The fact that even rudimentary information transfer between brains, even interspecies in some simple tests, is possible, has profound implications for understanding neural coding, maybe consciousness, and the potential for more sophisticated mind linking in the future. But the concerns are equally profound. Neural privacy within the net, agency, identity, the potential for coercion. It fundamentally challenges our concept of a biological individual.
These experiments really stretch the boundaries of possibility and force us to confront incredibly difficult questions about what it means to be an individual, about consciousness, and about responsibility when minds are linked.
They absolutely take the ethical discourse into uncharted, almost sci-fi territory.
Okay, let's zoom out to the biggest possible picture, the truly long-term view. If these kinds of neurotechnologies, augmentation, brain linking, were to become widespread, maybe even commonplace in some animal populations, what could the implications be? Not just for individuals, but for entire populations, ecosystems, maybe even the definition of a species.
If neurotechnology were deployed widely in animal populations, the source suggests the impacts could be profound and ripple far beyond individual animal welfare. We could see significant effects on behavior, ecology, and potentially the very integrity of species.
How might it change animal behavior and their social structures?
If you significantly alter an animal's senses, movement, or cognition via BCIs, it could drastically change their natural behaviors, how they find food, avoid predators, interact. This could severely disrupt established social structures within animal groups, which often rely on subtle, species-specific communication cues and hierarchies. If some individuals have technologically altered abilities or ways of communicating, it could affect everything from mating patterns and parental care to group cohesion.
Could it affect how animals learn from each other?
Potentially. Social learning and how information spreads through a group could be enhanced or distorted by BCI-mediated interactions. The source points out that human activities are already reducing behavioral diversity in social learning in many wild populations. BCIs could act as a potent and likely unpredictable new vector for modifying these fundamental processes.
What about the wider environment? Could BCI animals impact ecosystems?
Yes, that's a major concern. Introducing BCI-modified animals into the wild, or even widespread use in domesticated animals that interact with wild environments, could lead to significant unintended ecological consequences.
Gauss.
Altered foraging behavior due to enhanced senses could impact plant populations or prey species. Predator-prey dynamics could shift dramatically if one side gains a technological edge in hunting or evasion. Even subtle changes in how animals interact with their environment, like altered seed dispersal routes due to modified movement, could have long-term cascading effects on things like forest regeneration or plant diversity. The source notes we already worry about the ecological impacts of human-induced animal behavior change. BCIs represent a novel and potentially powerful way to cause such changes, perhaps with unpredictable outcomes, similar to introducing invasive species trophic cascades, ecosystem destabilization. Predicting these system-level effects is incredibly hard.
This sounds like it could actually interfere with evolution itself, changing the traits of animals in a population.
That's the most profound long-term concern discussed, the potential impact on species integrity and evolution. If you create animals with significantly enhanced cognitive or sensory abilities, or link brains across species, you start blurring the lines that define species' typical behaviors and capabilities.
It raises the question of what even defines a species anymore.
Exactly. And what value do we place on preserving a species' natural evolutionary path? The source draws an interesting parallel here to the concept of animal disenhancement.
What's that?
That's the controversial idea, sometimes discussed in the context of factory farming, of genetically or neurologically modifying animals to reduce their capacity for suffering, maybe making them less aware or less sensitive to pain or confinement. While often framed as a way to improve welfare in bad conditions, it's still a deliberate, artificial alteration of the animal's fundamental nature.
And neuroenhancement is the flip side.
Conceptually, yes, using BCIs to add capabilities. But it raises similar ethical concerns about the artificial modification of an animal's inherent constitution. You risk creating beings that are no longer truly representative of their natural species. And the ramifications could be huge and irreversible if, for instance, BCI-enhanced animals were ever able to interbreed with wild populations.
How might widespread use of this tech change our fundamental relationship with animals?
It could reshape it dramatically, gaining deeper technology-mediated insight into, say, our pets' minds might foster greater empathy and better care. That's the optimistic view.
And the pessimistic view.
The converse is that it could lead to increased instrumentalization. Animals viewed more as programmable entities, biological gadgets, components we can interface with and control. The source mentions the need for philosophical work on what a true animal interface would look like if it respected non-human experience. It also raises complex questions about our responsibilities towards animals we have technologically altered.
This goes so far beyond just helping a paralyzed monkey move an arm. It touches on potentially redirecting evolution, altering ecosystems, fundamentally changing our relationship with the entire animal kingdom. It's almost overwhelming to contemplate the possibilities, good and bad.
The potential for neurotechnology to act as this powerful artificial evolutionary force is significant. Unlike natural selection, which works slowly over generations via environmental pressures, tech could induce rapid, targeted changes within lifetimes. If those changes become widespread or heritable, humans could effectively steer animal evolution by design. This demands extraordinary caution and an ethically informed approach that considers not just individual welfare, but these much broader ecological and evolutionary consequences. The instrumentalization inherent in many proposed BCI uses, if scaled up, could seriously impact biodiversity and ecosystem health, bringing environmental ethics and conservation principles squarely into the neuroethics debate.
This deep dive really drives home that the conversation about connecting animal minds isn't just a tech fantasy. It has roots deep in our history and implications that stretch far into the future of life on Earth.
It forces us to confront our role and responsibilities as technologically powerful beings interacting with the natural world.
Wow. Okay, we have journeyed through an incredible amount of complex territory. We started with the basic science of BCIs, the tech challenges, the crucial history relying on animal research.
Then we looked at the ambitious goals, but also the massive practical hurdles facing ideas like a Neuralink for pets. We explored the long arc of animals and technology, the vital role of animal models in medicine, but also their limitations.
And we even touched on these really radical possibilities, neurotech as an evolutionary force, creating cyborgs and brainettes, blurring biological lines.
And underpinning all of that, we've wrestled with the profound ethical questions. The ongoing debate about animal consciousness and subjective experience, the sheer impossibility of getting true informed consent from nonverbal species, how we try to interpret signs of assent or dissent, and whether our current ethical rules like the 3 Rs are really sufficient for this new frontier of direct brain interaction.
The sources seem really clear on one thing. The idea of a rich, detailed, two-way mind connection between an animal and something like the internet, that's still very much science fiction for now. The technological, biological, and especially the interpretive challenges are immense and far from solved.
Absolutely. But the research pushing towards the capabilities that could eventually enable such things is happening right now. And that research is already raising critical questions we need to be thinking about before the technology gets much further along.
It really forces us to reflect deeply on our relationship with other animals, the value we assign to their minds and experiences, and the potential consequences, intended and unintended, of using technology to alter their fundamental being, from individuals right up to entire species and ecosystems.
And that debate around animal consciousness sits right at the center. Without a clear understanding or consensus there, the ethical justification for non-therapeutic, potentially mind-altering invasive neurotech becomes incredibly difficult.
Okay, so drawing this all together, here's a final provocative thought for you, our listener, to mull over, inspired by this deep dive into animal minds, digital bridges.
The sources highlight this key scientific goal: translating animal neural signals into information that humans can understand. Now imagine if we somehow achieved true success. Imagine we could genuinely understand an animal's complex thoughts, its emotions, its subjective experience through some advanced brain interface.
Would that breakthrough in understanding lead us, finally, to grant animals greater moral status, maybe even legal rights and stronger protections?
Or would our newfound ability to access and interpret their internal world simply make them more predictable, more quantifiable, more valuable as tools for human purposes, leading paradoxically to even greater instrumentalization?
That question really cuts to the heart of it, doesn't it? Would this ultimate connection bridge a gap in empathy and understanding, or just forge a new, perhaps more insidious form of control?
It's the central question hanging over the future of this technology and our relationship with the animal kingdom.
Thank you for joining us on this deep dive.
It's been a fascinating exploration.
If you are interested in content like this, we will discuss a topic that has intrigued us every day. Please be sure to subscribe to the channel and follow us so you don't miss future deep dives into complex and fascinating topics like this one.
See you next time.